Welcome back, my dear students. Today we'll uncover how neurons pass their messages across tiny gaps called the synapses. We'll see how electrical synapses allow signals to flow instantly while chemical synapses use neurotransmitters to bridge the space between cells. Step by step, we will understand how excitatory and inhibitory signals shape the communication process, decoding whether a neuron fires or stays silent. By the end of this class, you will have a clear picture of how our nervous system coordinates thought, movement, and response through this fascinating process of synaptic transmission. This is the image showing synapse. This is the gap or synaptic cleft between two successive cells. What happens during the signal transmission at synapse? A synapse is the junction where communication occurs between two neurons or a neuron and an effector cell, which is muscle or gland cell. The presynaptic neuron carries the nerve impulse toward the synapse that sends the signal, and the postsynaptic cell receives the signal. It can be a neuron or an effector cell. Types of synapses based on position: axodendritic, that occurs between axon and dendrite; axosomatic, that occurs between axon and cell body; and axoaxonic, occurs between two successive axons. So synapses are of two types: electrical synapse and chemical synapse. So what happens? This is the presynaptic neuron or presynaptic cell, and this is the postsynaptic cell. The gap between two successive cells are called as the synaptic cleft. What happens? In our last class, we have learned about the action potential. So action potential, when arrives at the action terminal, suppose this is the action terminal, so there voltage gated calcium channels open. This is the voltage gated calcium channel. When it opens, then calcium 2 plus enters the cell. From the outside of the cell, calcium 2 plus enters into inside the cell, and calcium 2 plus signals to vesicles. Calcium 2 plus ions, they send a signal to the synaptic vesicles. Then vesicles move to the cell membrane of the action or axolemma. Then docked vesicles, then there is docking protein which is responsible for docking of the synaptic vesicles on the cell membrane, and then the vesicles release their neurotransmitter which bound to the receptor protein present on the postsynaptic membrane of the plasma membrane of the postsynaptic cell. Then the neurotransmitter diffuses across the synaptic cleft and binds to the receptor. Binding of neurotransmitter to receptor then allow the ligand gated Na plus ion channel to open the postsynaptic membrane, allowing Na plus ions to diffuse by facilitated diffusion from the synaptic cleft. So what happens? From the synaptic cleft, the Na plus ions moves or enter inside the postsynaptic cell, into the postsynaptic bulb. If enough Na plus ions move into the presynaptic bulb, then another action potential will be activated at the action hillock. So we know that synapses are basically of two types: electrical synapse and chemical synapse. We'll discuss the two different types of synapses. So what is electrical synapses? It means communication occurs through gap junctions that directly connect the cytoplasm of two adjacent cells. Each gap junction contains connections, tubular proteins forming tunnels. Ions flow directly from one cell to another, so action potential spreads instantly. It is found in visceral smooth cell, cardiac muscle, embryonic tissue, and brain. Advantages of electrical synapses: faster communication, action potentials pass directly without delay. Synchronization, groups of neurons or muscle fibers fire in unison. That is example coordinated heartbeat or peristalsis in gut. This is the presynaptic neuron, this is the flow of ions, this is the gap junction. Electrical synapses occurs through the help of gap junction, but chemical synapses, there is no gap junction. They directly cross the synaptic cleft and enter into the postsynaptic neuron. So chemical synapse, here presynaptic and postsynaptic membranes are separated by the synaptic cleft, which is 20 to 50 nanometer wide, and it is filled with interstitial fluid. Impulses cannot jump directly, so neurotransmitters mediate communication. Here the steps of signal transduction at chemical synapses are following. Nerve impulses reaches the synaptic end bulb of the presynaptic action. Depolarization opens voltage gated Ca2 plus channels, then Ca2 plus enters presynaptic terminal. Increased Ca2 plus triggers exocytosis of synaptic vesicle, and then neurotransmitter released into the synaptic cleft. Neurotransmitter diffuse across the cleft and bind to receptors on postsynaptic membrane. Binding opens ligand gated ion channel. Specific ions flow in or out. Ion flow changes postsynaptic membrane or voltage, produces a postsynaptic potential. Depolarization, it is excitatory if Na plus channels open, or hyperpolarization, it is inhibitory if K plus flows out or chloride minus flows in. If depolarization reaches threshold, an action potential is generated in the postsynaptic neuron. So in this process, transmission is one way only, from presynaptic to postsynaptic. Example at neuromuscular junction. Here this is the presynaptic neuron, this is the synaptic cleft, and this is postsynaptic neuron. An impulse arrives at the end of the presynaptic neuron, then vesicles move towards and fuse with the presynaptic membrane. This releases neurotransmitter into the synaptic cleft. The neurotransmitter diffuses across the synaptic cleft down to a concentration gradient. The neurotransmitters attach to receptors on the postsynaptic membrane. This triggers an impulse which travels along the postsynaptic neuron. The neurotransmitters are recycled or destroyed once an impulse is sent. This is the postsynaptic neuron. So, what is excitatory and inhibitory postsynaptic potential? Excitatory postsynaptic potential, which makes the postsynaptic cell which excite the postsynaptic cell. It is caused by depolarization of the postsynaptic membrane. It brings the cell closer to threshold. It increases excitability. It brings the cell closer to threshold. It increases excitability, and then a single EPSP usually does not trigger an impulse, but multiple EPSPs can summit to reach the threshold. What is inhibitory postsynaptic potential? It is caused by hyperpolarization of the postsynaptic membrane. It makes the inside of the cell more negative and further from the threshold. It reduces the likelihood of action potential generation. This is the image showing EPSP or excitatory postsynaptic potential, which is a local depolarization of the postsynaptic membrane, and inhibitory postsynaptic potential is the local hyperpolarization of the postsynaptic membrane. EPSP bring the neuron closer to the action potential threshold, and IPSP drive them away from the threshold. So this is the diagram showing the excitatory synapse, and this is inhibitory synapse. So in today's class we explored how neurons communicate at synapses, whether through direct or the rapid flow of ions in electrical synapses, or the precise release of neurotransmitter in chemical synapse. We saw how excitatory signals push neurons close to firing, while signals hold them back, creating a perfect balance that ensures accurate control of body's function. This fine-tuned system of EPSPs and IPSPs is what allows thoughts, emotions, and movements to occur with remarkable precision. So always remember, every heartbeat, every idea, and every action depends on the seamless transmission of signals across synapses. Keep learning, stay curious, and I will see you in the next class. Thank you.